This presentation was created by Jessica Taylor, Jessica Batres, and Jasmine Chu. The topic we are covering today is upper motor neuron control of the brainstem and spinal cord. Upper motor neurons are essential for coordinating many different types of movements by signaling the circuits of the brainstem and spinal cord that innervate different areas of the muscular system. The types of movements that are generated by these pathways include posture, balance, voluntary movement, and locomotion. Some brainstem centers and cortical areas are where the upper motor neuron pathways begin. The sources of these pathways have great influence on the type of movement that will occur. Some pathways that we will go over in this presentation are the corticospinal tract, which contributes to movement of distal extremities, the corticobulbar tract, which contributes to the movement of the cranial nerves, the reticular formation and vestibular nuclear complex, which contributes to posture and position, as well as the primary motor cortex and premotor areas in the posterior frontal lobe, which contribute to voluntary movement. Discuss the descending motor control. Part A shows a cross-section of the spinal cord and the specific regions where the upper motor neurons descend through the spinal cord. Part B goes a little bit more in depth of what's going on in Part A. So we have the upper motor neurons in the cerebral cortex, which are the circled red dots, descending through the spinal cord through the lateral white matter, and they will be innervating lower motor neurons in the lateral ventral horn to innervate the distal extremities for skilled movement. The upper motor neurons in the brainstem will send an impulse to the lower motor neurons in the medial ventral horn through the white matter of the spinal cord, which will elicit posture, balance, or locomotion for the proximal extremities. In figure 17.3, we can see a cross-section of the primary motor cortex, and that cross-section includes cortical layer 5, where the pyramidal Betz cells and non-Betz cells are located. These cells are important because they make up the upper motor neurons that will connect to the lower motor neurons to control muscle activity. Betz cells are important for activation of lower motor neurons that control movement of distal extremities. And these are the largest neurons in the human body, but they only account for 5% of the axons that innervate from the motor cortex to the spinal cord. The remaining upper motor neurons are the pyramidal non-Betz cells, are found in the primary motor cortex and in the premotor cortex, where they descend into the corticobulbar and corticospinal tracts. Here we have a diagram that depicts the descending pathways of the corticobulbar and corticospinal tract that terminate in the brainstem and the spinal cord respectively, where they will innervate different nerves to elicit movement. On the top left, we have a cross-section of the motor cortex, and underneath we have cross-sections of the brainstem, which will include the midbrain, the pons, and the medulla. And after the medulla, we have a cross-section of the spinal cord. First, we will begin by discussing the corticospinal tract, which is depicted in dark red and light red in the diagram. So first, they will begin in the motor cortex on the more medial side of it and descend into the midbrain in a region called the cerebral peduncle after it passes through the internal capsule. It will then cross into the pons and descend into the medulla oblongata, where it will go into the medullary pyramid. After the corticospinal tract passes through the medullary pyramid, we can see here in the dark red portion that the majority of the axons in this pathway will cross over into the contralateral side of the ventral horn, creating the lateral corticospinal tract. This will innervate the movement of more distal muscles such as the fingers and the biceps. And the portion of the corticospinal tract that does not cross over is depicted in light red and is called the ventral corticospinal tract that innervates muscles such as the neck, shoulder, and trunk, which are more proximal, and therefore contribute to posture control. Moving on to the corticobulbar tract, which is a bit shorter than the corticospinal tract since it does terminate at the brainstem, since they only synapse with cranial nerves. The corticobulbar tract is depicted in the gold line on the diagram, where it will also descend into the internal capsule and go into the midbrain in the same region as the corticospinal tract, the cerebral peduncle, and then descend into the pons, where we can see that some of the axons of the corticobulbar tract branch out and synapse with cranial nerve 5 and cranial nerve 7. And following the corticobulbar tract down into the medulla, we can see that the corticobulbar tract branches off again and ends by synapsing onto cranial nerve number 12. It is important to note that most of the corticobulbar axons terminate bilaterally, which means that the muscles of the face are controlled by both sides of the brain. And this is helpful to reduce deficits in function when one side of the corticobulbar tract is damaged.
That was applied to the cortex, which is what allowed researchers to actually see the different areas of the brain that lit up, depending on where the electrical stimulation was applied. And this led to the discovery that the human motor cortex also contains a spatial map of the contralateral body. So musculature requiring fine motor control is represented by a larger area of the motor cortex than those of the trunk and lower extremities, which require less motor control. So as you can see from the picture, the areas from the trunk and lower extremity represent a super small portion compared to the upper extremity and the face. So this also shows how each of these movements are kind of connected through our brain. So even throughout the motor cortex, many regions that are responsible for movement overlap. So due to a study done involving monkeys, researchers were able to conclude that the primary motor cortex contributes to the initial phase of recruitment of lower motor neurons involved in the generation of finely controlled movements. The study with monkeys also led to the development of what is known as the muscle field. The muscle field is when activity of multiple different muscles is directly facilitated by the discharges of a given upper motor neuron. The average size of a muscle field is about 2 to 3 muscles per upper motor neuron. This concludes that activity of the upper motor neurons in the cortex controls movement rather than actual individual muscles. So now we're going to go on to the directional tuning of an upper motor neuron in the primary cortex. So as you can see in these pictures, the picture on the left is showing a monkey moving an object either forward, down, right, or left. And then the picture on the right is kind of showing how neurons are firing in those same directions. So as you can see in the figure 17.8a, which is the one on the left, the monkey's arms movement is being guided by a light. So what researchers did is the monkey would be moving an object to where this light would, to wherever the light would light up. And they changed this light every time to see how neurons fired depending on where the light was or if the monkey would be reacting to the light or the movement. So because of this, researchers were able to predict the monkey's arm movement using neuronal population vectors. This is based on the fact that motor neurons fire in directions. So the picture on the right, which is figure 17.8d in the book, shows the discharge rates, which is shown by the black lines, prior to direction of movement. So as you can see, it's not just one solid line, it's a bunch of different spikes, and they're all at different lengths. And this is because so many motor neurons fire per individual movement. They are considered too broad to determine direction of movement, however, so this is why it is important to always look at starting position and then final position. To the premotor cortex, which is shown in this picture as being represented by the blue regions, and the purple is what we just talked about, which is the primary motor cortex. So the premotor cortex is best understood in studies done with macaque monkeys, and up until recently, with studies done on patients presenting with frontal lobe injuries, we have just now been able to conclude that human brains might actually have the same premotor areas that we have always seen in the macaque monkeys. So upper motor neurons in the premotor cortex influence motor behavior both indirectly and directly. It is represented indirectly through extensive reciprocal connections with the primary motor cortex and directly through axons that project through the corticobulbar and corticospinal pathways to influence the local circuits that organize the output of lower motor neurons in the brainstem and spinal cord. So over 30% of the axons in the corticospinal tract are actually from neurons in the premotor cortex. However, there's one major difference between the premotor cortex and the primary motor cortex, and this is what's most important. So action goals encoded by the motor cortex are typically localized to personal space, whereas actions encoded by the premotor cortex are oriented towards extrapersonal space. So what we mean by the personal space is like from the shoulder to the elbow, and the extrapersonal space is located before that. So more than 50% of the neurons in the premotor cortex are linked in time to the occurrence of movements, and I actually believe the percentage is around 65%. And most of the neurons that fire are firing before and during a movement, and they're all made in one direction. So these are considered closed loop. In the motor cortex, neurons don't fire until the appearance of a cue. So going back to the study with the monkeys, the cue would be when the light turned on, or when a researcher is showing the monkey what to do, that cue is picking up the object or moving the object to a different direction. So this is kind of where scientists got our mirror neurons from. So mirror neurons are associated with the premotor cortex, and how this works is that when a monkey sees someone else performing an action, action potentials are fired in the premotor cortex, and these same action potentials are fired when the monkey is actually performing that same movement itself. So when we look at this picture, you can see that when the monkey is just at rest, its mirror neuron is also at a resting state. There's no electrical signals firing that any computers are picking up on. And then as you can see in the B portion of this picture, the monkey is grabbing the object and the mirror neuron is firing, and you can see that it's electrical signals at the bottom represented by the three little humps. And then in the picture C, the monkey is not actually moving the object itself, but it's watching a researcher move the object. And if you look at where the mirror neurons are firing, it's represented by the same exact movement as when the monkey was actually grabbing the object itself. And this is where we briefly move into our last section of the premotor cortex, which is the Broca's area. And this area is a rostral division of the lateral premotor cortex that has evolved to play a role in speech sounds, which is super important. So anything to do with us reacting to noises or making noises is all part of this Broca's area.
Within the brainstem, there are the vestibular nuclear complexes and the reticular formation and superior colliculus. The reticular formation is made up of clusters of neurons or axon bundles for sensory processing of the temporal and spatial movement, coordination, and locomotion of the body. Functions for skilled voluntary movements and motor reflexes include movements ranging from cardiovascular control, sensory motor reflexes, eye coordination, alertness, and coordinated speech. For reticular formation, it is also considered a net-like term within histology, located in the rostral midbrain to the caudal medulla. In the figure to the right, we can see bundles of neurons of the reticular formation within the brainstem of the midbrain, lower pons, and middle of the medulla. First, we have the mesencephalic reticular formation and rostropontine reticular formation on the top and assisting the coordinating brain activity in the forebrain, whereas the caudopontine and medulla reticular formation assist in the premotor coordination of lower somatic and visceral motor pathways, either through our senses or through our voluntary motor movements. On the right, we have the reticular formation pathway, or the reticulospinal tract, going from the spinal cord to the brainstem up to the premotor cortex on the cerebrum. At the area where it says reticular formation, around the middle area, there are the indirect projections and the direct projections going down the motor cortex to the spinal cord. For the reticulospinal tract, we have indirect and direct projections. The indirect projections primarily control the motor muscle fibers of proximal muscles as information is sent from the brainstem, for example, muscles in the neck and abdomen. For the direct projections, motor cortex controls the speed, precision, and agility of movements. For example, when using your hands to grasp something or using a remote control. As part of the vestibular nuclear complex, there are two pathways to maintain posture from anticipated or compensatory feedback, caused by either postural instability, which goes through the vestibular labyrinth, or it can be feed forward from external stimuli in order to stabilize posture during ongoing movements, such as sports or walking or running. The lateral and medial vestibulospinal tract both innervate through the anterior white matter in the spinal cord. For the lateral vestibular tract, it is positioned more ipsilateral to the medial vestibular tract and the medial lower motor neuron pools, which will lead up to the lateral vestibular nucleus. In this tract, the proximal limbs muscles are stimulated to signal deviations for balance and upright posture. Within this local circuit of neurons, the cranial nerves control eye and head movements. So if you were moving your head but fixing your eyes on a static object to your right, then you would use your lateral vestibulospinal tract to focus on that object with your eyes while moving your head simultaneously in the opposite direction. For the medial vestibulospinal tract, the cervical spine is innervated through the medial ventral horn toward the medial vestibular nucleus. This tract helps reposition the head when unbalanced or unstable by activating the neck muscle reflexes. For instance, if a person or infant is leaning forward and about to fall, the medial vestibular tract will instigate a reflex activation in order to remain or return to a balanced state, similar to how a rubber band works when you bend it forward with gravity and sling it back. In the third upper motor neuron pathway, we have the superior colliculus. Here it is located in the rostral to the reticular formation and is dorsal to the midbrain of the upper motor neuron pathways. This image is also seen in the beginning when we went over reticular formation on slide 11. So here we have the two neuron pathways, the colliculospinal and tectospinal tract. Both of these tracts work together to control the lower motor neurons in the spinal cord by innervating neural circuits to assist the reticulospinal projections in the neck. And similarly, these projections are medial cell groups within the cervical spinal cord, which allow for control of the axial musculature in the neck in order to orient and position the head and eye movements. The upper motor neuron pathways also are compiled of syndromes, a set of degenerative nerve damage of the descending motor pathways. This can lead to damage in the motor cortex of the brain or at local areas of the contralateral muscles. Some of the signs include hyperactive reflexes, such as clonus, spinal shock, impaired voluntary movement, and spasticity. First, we have spinal shock, also known as hypotonia, which is motor neuron damage, which decreases input from motor cortices in the brainstem. This includes the Babinski test, spasticity, and the loss of ability of fine motor movements. The Babinski sign is caused by damage to descending motor neuron pathway of the peripheral system, which is tested at the sole of the foot. The clinician would stroke the middle of the sole of the foot from the heel to the toes, also seen in the figure on the top right, and stroked from the heel up to the toes. If the toes flex downward, then it would result in a normal plantar flexion response. But if the toes fanned out into a plantar extension or hyperextension, then it would result as the Babinski sign, as there is damage in the descending upper motor neuron pathway. Spasticity is caused by disrupted motor cortex of the vestibular nuclei and reticular formation, and is often described as an increase in muscle tone or a hyperactive stretch reflex, often known as clonus. Damage to the vestibular nuclei often causes damage to the corticospinal tract, leading to increases in spinal cord stretch reflexes. Lesions in the descending pathways damage the lower motor neurons that connect to distal upper limbs, such as the fingers, in order to make precise fine movements. This may also occur over time due to deterioration or damage of neural pathways. Lastly, decerebrate rigidity is similar to spasticity, where the descending pathways may become damaged but in higher regions of the brain, above the vestibular nuclear complex or in the medulla oblongata. The body will become stiff and rigid, similar to being stuck in a full extension, having a clenched jaw and retracted neck due to the limited neural stimulation to that area of the brain.